Okay, in this video, I'm going to go over chapter 4, which covers the bacterial culture, growth, and development. Okay, so this is the outline for the lecture. We're going to go through microbial nutrition, then nutrient uptake, so how they acquire nutrients. We'll talk about how to grow bacteria and count them. We'll talk about the growth cycle, biofilms, and then cell differentiation. Okay, so let's talk about how bacteria kind of grow and obtain their biomass. So when we talk about biomass, we're really talking about how the bacteria obtain carbon, because carbon is kind of the main substance that most organisms need to acquire. And so there's two types. There's heterotrophy, which means that they obtain their carbon from some kind of organic material. In the diagram, they're showing it as organotrophy, but you'll see that organotrophy also means something else. But really, heterotrophy just means that you're getting your carbon from some kind of organic material, whereas autotrophy means that you're getting your carbon from CO2. And so you get CO2 and you fix it, usually through like the Calvin cycle, and then you produce your organic molecules from there. So you're taking it from the air, essentially. Okay, and that's just the carbon, acquiring carbon. Organisms also need to acquire energy and electrons. So now we're going to talk about that. And so there are a bunch of other vocab words that have to do with how they acquire energy and electrons. So I kind of like this diagram. The diagram is not in your book, but it kind of does a good job of summarizing the trophies, I guess. There's a lot of troughs. And so phototrophy, photo means light, and so the organism is getting their energy from the sun, whereas chemotrophy means that they get their energy from chemicals. So usually that's like breaking down glucose into energy or something like that. And then there's also the electrons. So organotrophy means that the organism is getting their electrons from some kind of organic material, something that is a lot living or dead, but something that was at least an organic substance. Whereas lithotrophy means that the organism is getting its electrons from rocks, and so, or some kind of non-living thing. But yeah, I like this diagram because it kind of summarizes the photo and auto, chemo hetero, and photo hetero trough, and it kind of gives you a summary of at least the energy and the carbon source. Not, it doesn't include the electron source, but it's still a nice diagram. Okay, so now that the organism has acquired energy, it needs to store that energy. And just like most organisms, it stores its energy through ATP. So ATP is adenosine triphosphate. You should know this. And organisms use it to store energy. And then, of course, the ATP can be used to power different types of chemical reactions. Okay. So the kind of enzyme that is used to make ATP is called ATP synthase. Synthase means to create. And this ATP synthase is powered by proton motive force. So there's like a concentration gradient of hydrogens on one side, and there's not a lot of hydrogens on the other side. So the hydrogens move from high to low concentration. And when the protons or hydrogens flow, they will go through this F0 rotor, okay? And when they go through the F0 rotor, it causes this gamma stalk. So the gamma stalk is down here. It causes it to rotate. And this rotation causes the production of ATP. So that's in this third step here. So the rotation of the actual structure causes ATP to be created. So very similar to how like a windmill creates energy, the movement of the propellers in the windmill help generate energy. Okay, like we've talked about already in the class, bacteria are very important for the nitrogen cycle, and we'll talk about this again later on, but this is just kind of a deeper dive into the nitrogen cycle. So bacteria are responsible for 3 types of reactions in the nitrogen cycle. So the first one is nitrogen fixation, where we're getting nitrogen gas and converting it to NH3 and NH4 plus, so basically ammonia. And so that's the nitrogen fixers. Now there's certain types of bacteria that do this. Now that we have ammonia, we can also go through nitrification. So other types of bacteria can get that ammonia and convert it to NO3 minus, which is nitrate. Okay. And then denitrification is basically getting that nitrate and converting it back to N2. So it's the whole cycle. And again, different types of bacteria do the different roles. So it's not like one bacteria can do all of this. Different types of bacteria do the different parts of the cycle. Okay, so that was it for 4.1. Now we're in 4.2, where we're talking about how bacteria take in their nutrients. Like physically, how do they actually take the nutrients into themselves? So there's three ways that they can do this. They can use carrier proteins like this that physically, oh, actually, this isn't a carrier protein. Carrier proteins that physically bring the molecule inside. Then there's these nutrient binding proteins that either bind to the nutrients on the cell wall or periplasmic space, but anyway, there are proteins that will bind to different types of nutrients. And then the last one are channel proteins or pores. This is a channel protein. Basically, it creates a pore on the cell membrane so that nutrients can go through the pore and get inside of the bacteria. Okay, and so we're going to talk about all of the different ways that bacteria take in nutrients. So let's first start with the channel proteins or pores, because this type of transport is what we call passive transport, meaning that the solution or the molecules are going down the concentration gradient. So they're going from high to low concentration, which does not require energy. So that's, again, called facilitated diffusion, if you haven't heard it before. So these channel proteins, again, they create a passageway for these molecules to go through, but we are not using energy because we're moving the molecules from high to low concentration, which is moving down the gradient. And so here is glycerol. There's a bunch of glycerols on the outside. This channel called GLPF is this molecule that provides the space for the glycerol to go inside, and that's pretty much it. And again, no energy is involved. The glycerol just flows in. Okay, but for most situations, when you think of bacteria, they're trying to take in nutrients in a situation where there are not a lot of nutrients outside. So for example, if you're a bacteria like living in a river or a lake, there might not be that many nutrients out. And so that's going to be a problem because it's going to be very rare for the situation to have high nutrients on the outside and low nutrients on the inside. It's very rare to have this concentration gradient where it's going to go from high to low like this. Usually it's the opposite. And when it's the opposite, we have to use energy because we're going against the concentration gradient. So we're going to talk about active transport. And the first ones are symport and antiport, which together are the types of coupling transport. And basically what this means is that two molecules are being moved, two types of molecules are being moved at the same time. So with both of these, you're going to have one molecule that moves down the concentration gradient, again, from high to low, and that generates energy. And then the energy that has been generated then is used to move a different molecule from low to high concentration. So an example of this is symport, where both molecules move in the same direction. So let me kind of zoom in here. So what's happening here is that there is a high amount of these red molecules on the outside and low inside. And then there's a high amount of blue on the outside and there's low amounts on the outside. Okay, cool. And what we're trying to acquire is the blue one. That's really our target. The blue is, for example, glucose. So we want to get in glucose, but to bring glucose in, we're going to be moving the molecule from low to high, which needs energy. And so the energy that we're going to get is from the other molecule. So this molecule is in the high to low situation. If it were to move inwards, it's going from high to low. And because that generates energy, that energy is used to bring this blue molecule from low to high. Okay, so one molecule moves from high to low, and this molecule moves from low to high. And together they move inwards into the cell. And so that's called symport because they're both going in the same direction. They're both going inwards. But again, one molecule is going from high to low and the other molecule low to high. Now, the other one is antiport, which is essentially the same principle, except we are going in the opposite direction. So let me zoom in here. So here in this situation, we want to bring the blue molecules in. And again, it's going from low to high, so that would require energy. Now, there's a lot of red molecules on the inside and not a lot on the outside. So if we were to push the red if we were to allow the red molecules to go from high to low, that would power the movement of the blue molecules from low to high going inwards. So again, it's the same principle. The red molecule is going from high to low, and the blue molecule is going from low to high. So that's the same as in symport. The only true difference is that they're going in opposite directions rather than the same direction. So here in the symport, they're both going inwards. Here in the antiport, one is going out and one is going in. But the principle is the same. The red one is going from high to low, whereas the blue one is going from low to high in both situations. Okay, cool. And then, of course, that is just a very specific type of situation. Most of the time, we are going to actually need to use ATP. And so there are these proteins called ATP binding cassettes because they use ATP to move things. And we have shortened that to ABC. So ATP binding cassette is just short for ABC is short for that. So we call these ABC transporters because they use ATP to move molecules against the concentration gradient. And the way it works, very simple. So the molecule that we want to move will bind to the transporter, which is the protein on the membrane. And then there are these ATPases, which are enzymes that break ATP. And when the ATP breaks, the energy is released and then the molecule is moved. That's pretty much it. And again, we are going from low to high concentration. We're going against the gradient, so you do require energy, and in this case, the energy is directly ATP. Cool. Then we're going to talk about iron uptake. So iron is very important for bacteria. They don't get a lot of it, but they do need it. And so they have a whole system of trying to acquire iron. So they use these molecules called siderophores. Siderophores are these molecules that are secreted by the bacteria, and they basically go out and hunt for iron. So the way I like to think of siderophores is like when you're fishing, right? When you fish, you have like the fishing rod and you whatever, cast your fishing rod, whatever I'm talking about. You know what I mean. You're letting the hook fly into the water and then you're fishing for fish, right? So that the fish can come. So a siderophore is kind of similar to a fishing rod where you're sending something out to acquire iron and then you're going to bring it back in. Okay. So the way that the siderophores work is that they are secreted outside of the bacterial cell and they will bind. So here's a siderophore, it's like this, whatever shape this is, hexagon looking thing. And this thing is going to find iron 3+, and when it finds it, it will come back. Doesn't really come back, right? It's not like an animate object or anything. It's an inanimate object, so it's not like it knows to come back, but eventually it'll, sit around near the bacteria, and then it will be guided back into the bacteria. And the way that it re-enters the bacteria is by going through the outer membrane first. It's going to go through this outer membrane channel. And then to go through the inner membrane, usually we will need some kind of energy source. And so we will use some kind of ATP powered protein, usually an ABC transporter. Will bring this entire siderophore inside. And then now we have the iron, and that's awesome. And then the siderophore gets reused because really the whole point of the siderophore is just to grab the iron, right? So just like your fishing rod, you can reuse the same fishing rod over and over again. So, yeah. All right, cool. The other type of kind of acquiring energy or acquiring molecules is through what is called group translocation. And what that means is that, or like, how does this work? Is what we're doing is when the molecules come into the bacterial cell, there is a functional group that is added. That's why it's called group translocation. So a functional group. So the functional group could be like a methyl group, or in this case, phosphate. But whatever kind of functional group is attached to the surface of the molecule that we're trying to acquire. And why is that helpful? Well, remember, we're talking about concentration gradients. So for example, if we want to take in glucose, there's very little glucose on the outside and there's a lot of glucose on the inside. So we're going against the concentration gradient. If we bring in more glucose, we're making that concentration gradient even worse, right? Because now there's even more glucose on the inside. So that makes it even harder to bring more glucose. So every time you acquire a nutrient, you're making the concentration gradient worse for yourself. But if you add a functional group, that isn't technically glucose anymore. So for example, if I were to add phosphate to my glucose, instead of it being glucose on the inside, is now glucose phosphate. And that is not the same as glucose, and so it does not interfere or does not create a concentration gradient. So the intracellular concentration of the molecule that you desire, for example, glucose, does not actually quote unquote increase. I mean, technically you are increasing it, but in the sense of the chemistry world, it is not being increased because glucose is on the outside, but on the inside it's glucose phosphate, which is totally different from glucose in terms of chemistry. So there is no quote unquote concentration gradient being created. Okay. And so this is just one example. It's called the phosphotransferase system. So either glucose or mannitol, which are two types of sugars, when they go inside, they will be phosphorylated as they enter. So you'll produce glucose 6-phosphate or mannitol 6-phosphate. And so if you were to look at the concentration gradient, you have a high amount of glucose 6-phosphate, not a high amount of glucose. And so that means that you don't have a low to high, quote unquote, concentration gradient of glucose. You have a low to high concentration gradient of glucose 6-phosphate. And again, in the chemistry world, if you add a functional group on top of a molecule, it's a different molecule altogether. And so it doesn't count towards the concentration gradient. And so then you can still continue to acquire more glucose without it being harder. If we were just taking in glucose after glucose without phosphorylating it, you would eventually have 1,000 glucose molecules on the inside and maybe only 10 on the outside. And those last 10 are going to be impossible to bring in. It would take a lot of energy to do that. But if you have 1,000 glucose 6-phosphate on the inside and 10 glucose on the outside, well, it doesn't matter because they're not technically the same molecule. That's how this works. Okay. All right. So now that we're done with talking about movement of nutrients, let's talk about growing bacteria in the lab. This I'm going to go over very briefly because a lot of this you're going to be learning in the actual lab portion of this class, so I'm not really going to go crazy. But to grow bacteria in the lab, we can grow them through streaking. So on a petri dish, you can streak the bacteria where you're basically diluting them out until you can get these colonies. And the reason why we like colonies is because a colony originates from an individual bacterial cell. So we're basically spreading the bacteria out until we have individual cells, and those individual cells grow into colonies. And so the theory is that one colony is basically a cluster of bacteria that are all identical to each other, which is very helpful when you're working with them in the lab because you want to be working with pure cultures. Another way that you can do this is through dilution. So we'll talk about this in the lab also, where you're basically diluting out the bacteria until you can get, again, individual colonies. Okay. When we grow bacteria, there are different types of media. So media is just a general term for any kind of substance that grows microbes. And so there is complex media. Complex means that the composition of the media is unknown, meaning that it's very rich in nutrients, but you don't actually know what's in there exactly. So the way I like to think about this is if you're making a soup, right? Like actually making soup at home, you get some like meat and you throw it in the pot and you get some vegetables and you throw it all in there. It's very rich in nutrients, but if somebody came up to you and asked you how many calories is in here, how many what's the milligrams of salt or what's the percentage of whatever, you would not be able to tell them because you just threw in some food into a pot, right? So that's the same concept with complex media. We're basically just getting, beef broth or some kind of yeast extract and just kind of shoving it all in there. We know that it's very nutritious, but we don't know the actual composition. Now, the opposite of that is synthetic, which means that we do know the exact composition. And so usually these are substances that are made, these are media that are made from pure substances. So the analogy that I like to use is, if you use pre-workout or anything like that, vitamins and like vitamin supplements, if you were to read the back of the label in your pre-workout or your vitamin supplement, they literally can list out every percentage of protein or every percentage of vitamin D, blah, 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 right? And the reason why they can do that is because when they created that vitamin or pre-workout, whatever supplement you're taking, they're using pure substances, right? They're using pure vitamin D and mixing it in there. They're using pure protein, whatever the heck you're taking, but all of those are pure substances, so they know the exact composition of the thing that you're consuming. Okay. Then we have media that is selective, and as the name kind of suggests, we are selecting for only certain types of bacteria. So we're just making sure only one type of bacteria or a certain type of bacteria can grow. And then differential means that different bacteria look different. Like literally they look different on the media. So this is an example of a type of media where the lactose fermenting bacteria are like darker and the non-lactose fermenting ones are lighter. So that's just an example of differential media. Now, most bacteria are really easy to grow in the lab, but there are some that need what are called growth factors, meaning that they can't just grow on regular, media. They need to have these specific nutrients. They're very like picky, I guess. So, for example, I'm just trying to look for one that's like a good example. Like Staphylococcus, they need to have, never mind, that's not a good example. Streptococcus, they need to have glutamate and alanine, like those are specific types of growth factors. If you don't supplement them with those things, then they're not going to survive. Or Mycobacterium, they need nicotinic acid. So, some bacteria just need these extra supplements to be able to grow in the lab. And then there are some microbes that we call the unculturables, meaning that we still have not figured out how to grow these bacteria in the lab. We know that they exist. We've done the DNA sequencing, but we just don't know what specific growth factors they need. So they're still considered unculturable. We don't know how to grow them yet. And maybe we'll never figure it out. Okay, so now we're going to talk about counting bacteria. This we do all the time in the lab because we want to know how many bacteria there are. So there's the direct way, which is just you literally counting them. So you look at them under the microscope and you count. So that's that. You're just counting. Another one is using a machine, so fluorescence activated cell sorting. And and by the way, there's like a shorthand for this. So FACS. So you'll see that acronym in the textbook. But anyway, so this basically has a machine that counts the cells. The cells have a fluorescent tag. The cells go through a machine. The machine counts them. That's pretty much it. And there's an indirect way of counting bacteria. This doesn't actually give you an exact number, but it tells you a relative idea of how many bacteria there are, and that's using optical density. So we already talked about how when bacteria grow, they make liquids cloudy. The liquid becomes cloudy when there's bacteria inside of it. So we use a spectrophotometer, and I'm sure you've experienced a spectrophotometer in chemistry, but it basically measures absorbance, right? And so we use the spectrophotometer to measure how cloudy the liquid is. So obviously, the more cloudy, the more bacteria. That's pretty much it. Okay, moving on to the bacterial growth curve. So now we're on 4.4. Bacteria, when you grow them in the lab, they follow this growth curve, especially if you give them, if you put them in one container and, you give them this amount of media, then they're going to go through this growth curve. So the first part of the growth curve is called the lag phase. Basically, the bacteria are getting used to their environment. And the lag phase can be different depending on the bacteria. It also is different depending on where they came from. So for example, if I was growing bacteria in a very nutrient-rich environment, and then I suddenly put them in a very nutrient-depleted environment, they're going to have a very long lag phase because they're going to be pretty shocked when they're like, oh my God, there's no more glucose. Like, what happened, right? So the lag phase would be very long. Now, if I were to have bacteria that were growing in a very nutrient-rich environment and then move them to the same nutrient-rich environment, just a fresh batch, their lag phase would be very short because it's basically the same environment, you're just replenishing the nutrients. So they're going to have a very short lag phase there. So that just depends on what situation you're putting the bacteria in. Okay. Then we have the log phase, also known as the exponential, because the growth is exponential. The bacteria are doubling over and over and over again, and the number of bacteria increases exponentially. Okay, so this is the happy time for the bacteria. But then once you reach a really high number of bacteria, they're going to start running out of nutrients, and that's where we get to stationary phase. So the cells stop growing, or some of them start dying and some of them are still growing. And so it's kind of like a balance. And so in stationary phase, it's kind of flat where the cells are starting to stress out. They're like, okay, we're running out of nutrients. We're kind of slowing down our growth. So like, what are we going to do? And then if you leave them in this environment, they're going to obviously run out of nutrients completely. And so then they're going to go through the death phase where they literally start to die. And again, this happens because they're just out. They're out of nutrients. They don't have any resources left. Okay, cool. There is some math involved with the growth of bacteria. So I'm just going to go over these two equations with you. So the first equation is this one, and I'm going to go over the variables. So NT stands for the number of cells, final, and NO stands for the initial number of cells, and then N is the number of generations. So I'm just going to give you an example of how you can use this. Let's say I wanted to figure out, how many cells, if I started with, let's say I started with 5 cells, okay, and I allowed them to double, because when bacteria grow, every generation is a doubling, right? Every time a bacteria divides, it just doubles. So let's say that I've doubled it 3 times. So I'm trying to figure out what the final amount of cells there are. So we started with 5, and there were three divisions, basically, meaning that we allowed them to divide 3 times for 3 generations. And so what is our final concentration? Well, that's 5 times 2 to the power of 3. 2 to the power of 3 is 8. So 5 times 8. Yeah, and that would be 40. Okay. And so that's just an example of how you would use that. So it'd be a very simple word problem where I'm like, okay, we're starting with this number of cells. We're having this many generations or this many divisions. So you just plug that in and then you've solved. Okay. The other equation is G equals T over N, or T divided by N. And so T is time and N is still the same, the number of generations. And then G is your generation time. So generation time means the time it takes to divide or double / double. Meaning, if I had 5 bacteria, how long would it take for me to then have 10 bacteria? So for the number of cells to double. Okay, or in other words, for the cells to divide, because if I had 5 bacteria and they all divided, then I would have 10, right? And so what is that time? How long does it take for a bacteria to go from 1 to 2 that's essentially the question. And so all you really have to do is plug in again. If, for example, I told you that it took 120 minutes for 3 generations, 3 generations, I'm just going to put gen, then you just do G equals 120 over 3, right? Yes, all you're doing is plugging it in. And then it's what, 40? Yeah. 40. Oh, 40 again. That's kind of weird. I did not do that on purpose. I'm just making up the numbers in my head. But yeah, 40 minutes would mean that it takes that bacteria 40 minutes to divide. Cool. E. coli, it typically takes 20 minutes. So it would be something like 120 over 6, right? Would be a 20-minute generation time. But yeah, very simple equations that you're plugging into. Okay, so now we're going to move on to 4.5, where we're talking about biofilms. So biofilms is a situation where bacteria can actually live in a community together, which is very, very cool. So this happens specifically when there's a lot of nutrients. The bacteria are super happy. And usually there are other situations where they're not happy, but for the most part, bacteria will form biofilms when they're in a nutrient-rich situation. And what will happen first is the bacteria will start to attach themselves to some kind of surface. And then the bacteria start to grow in a colony together. We call it a microcolony. And here in the microcolony, they start to communicate. They are able to actually communicate with each other through quorum sensing. We'll talk about quorum sensing more later, but essentially they're sending molecules, signaling molecules to each other. And a lot of these molecules are telling each other like, hey, I'm here. There's like 8 of us. I don't know, like they're just communicating how many there are. how many of themselves there are. Okay, and then they start to cluster up and they will make these exopolysaccharides. Basically, polysaccharides are secreted and it forms this protective layer around the group, which is very nice because biofilms are actually very resistant to things like antibiotics and disinfectants. They're really hardy. Okay, and then they start to grow even more and more, and you get this three-dimensional biofilm where there's like a bunch of bacteria living together in one colony or one community. And then if there is some kind of stress, like starvation or whatever, then the biofilm breaks and the bacteria disperse and, they try to escape the situation. Now, if they find another place that's very nice, then they will colonize over there. Or they will die. So that's the only two options that they have. Now, examples of biofilms would be like the plaque on your teeth, right? So plaque in your teeth is a biofilm. That's basically just bacteria literally caked onto your teeth, which is why it's good to brush your teeth. Because eventually those bacteria that are living there in the plaque will create these acids, and the acids start to degrade your enamel, and that's how you get a cavity. So yeah. Cool. That's all I really have to say about biofilms. They have like a bunch of other things in the textbook about like how they differentiate and all that, but this is pretty good. Okay. The last part of this chapter is on differentiation, and so we'll first talk about endospores. So differentiation means that the bacteria become different from their regular form. So they have like their normal form, and then they have like a weird different form. And so endospores are one. Basically, it's a dormant state that these bacteria can enter, and when they're dormant, nothing can really harm them, and they usually do this during times of stress. Now, I want to mention that not all bacteria can form endospores, only a few. And again, these endospores are very protective. They don't, they're really good at being protected from UV light, from being dried out, antibiotics, all this stuff, disinfectants, they're really protected. But they are dormant, meaning that the bacteria are not actually like dividing or actively growing. Now, if the conditions get better, then the bacteria can go back into their vegetative state. So yeah, that's good for them. Okay, so let's just really quickly go over the stages of the endospore formation. So we have the first stage where the bacteria decide, okay, I need to form an endospore. So what happens is there's a septum that forms. Okay, but notice that it's not symmetrical. One side is smaller than the other. And then the DNA starts to replicate. So DNA replication happens. And then in stage 2, some of the DNA goes on one side, some of the DNA goes on the other side. And then they completely split. Okay, notice that there's still one unit, but there are technically now 2 cells. We call it the mother cell and the forespore. Okay, and again, there's 2 copies of the DNA so far. Now in stage 3, the mother eats the child, basically. So the mother cell engulfs the smaller cell. So now there's a cell inside of a cell. Kind of weird. And when that happens, the DNA of the mother cell gets degraded. So now we only have 1 copy of the DNA. In this one, the DNA is still being degraded. Okay, great. Then the spore starts to produce, well, not the spore, but we start to coat the spore with a bunch of proteins. Okay. So things like dipicolinic acid start to form, calcium. There's a bunch of things that coat the spore. Basically, we're trying to give as much protective layers around the spore as possible. And then at one point, the spore is released. So now it's just all alone. Okay, the mother is gone. It's just the spore kind of naked. And then that's it. And the spore will stay there and can be, can survive for several years, honestly, in this state. Now, if the conditions are right, then the spore will go back to its vegetative form. And that's how it happens. Or it will go back here and then it's like, never mind, I want to go back. And so it can go back here. But for the most part, a spore will only germinate, quote unquote, if the conditions are good, because then it will go to the vegetative state and then it starts to grow like a regular bacteria. Pretty interesting. Okay. The other type of differentiation is in cyanobacteria. So cyanobacteria are these photosynthetic microorganisms, and they live in the ocean and water primarily. And so just like how we were talking about the nitrogen cycle, nitrogen fixation is very important because the plants acquire the ammonia from the nitrogen fixing bacteria. Now, cyanobacteria are going to be in trouble because there aren't that many nitrogen fixing bacteria that are kind of in the water. And so they have to fix their own nitrogen, which is really troublesome. And so they have these specialized cells called heterocysts that will fix the nitrogen for them. So it's still a cyanobacteria. So this is a cyanobacteria. This is also a cyanobacteria. So all of these cells are cyanobacteria. It's just that these are specialized cells that their primary job is to fix the nitrogen instead of doing photosynthesis. So why does this happen? Why can't they just do nitrogen fixation and photosynthesis in one cell? Well, the cyanobacteria, when they do photosynthesis, it produces oxygen, as you know. Photosynthesis makes oxygen. And it happens that the enzyme that fixes nitrogen is very sensitive to oxygen. So those two things could not happen in the same location. You can't have nitrogen fixation happening at the same time as photosynthesis or even in the same area. And so what the cyanobacteria have figured out is that some of these cells are responsible for photosynthesis, and then every once in a while, one of these cells differentiates into a heterocyst, and its only job is to fix nitrogen. Again, this is very unique because when you think of bacteria, you think, oh, they're all the same. All E. coli are the same. All cyanobacteria are the same. But here they are differentiating almost like how our cells in our human body have different functions, right? The cells in your stomach have a specific function that is different from the cells of your skin, for example. And so it's very interesting to see bacteria do this because usually we think bacteria, they all just do the same thing, right? But here you can see that they are living in one unit together. Some of them are photosynthetic and some of them are the fixers, the nitrogen fixers. Pretty cool. Okay, moving on, these are Myxococcus, specific type, very cool type of bacteria that can differentiate also. So they are able to move around through this thing called gliding motility. So gliding motility is this way that they can kind of move around on a surface. And they can do this either individually or they can do this as a swarm. So here you can see that they are swarming and they're literally crawling together on this surface. Really kind of weird. And then if they want to, during starvation, they will actually clump together. So here in the 72-hour image, they have clumped together to form this, what is called a fruiting body. So it's super weird. Okay, and then the last type of differentiation I want to talk about is filamentous bacteria, or bacteria that form these filamentous structures. By the way, filamentous structures are typically known in the fungi world, so mold, mushrooms, those are the ones that produce filamentous structures, so it's very rare to see it in a microbe, in a bacteria specifically. So there are some bacteria that can do this, where they form these mycelia, and mycelia are these filamentous strands. So all of this are mycelium, okay? And it's almost like roots, but not really. They're like these really small strands that are either in the soil or nutrient, whatever they're growing on. So that's pretty weird because bacteria typically don't do that. And then when they have nutrient depletion, they will start to grow aerial mycelia. Okay, aerial mycelia meaning that it's in the air rather than under the ground. Now, if the conditions are good, then these aerial mycelia, if there's like a high amount of glucose, then they'll go back to the original form, and that's cool. But if it's really bad, let's say that the situation is really unpleasant, they're going to go through this where the aerial mycelia get super long, super long, okay? And they'll eventually form sporangia, where there are spores. So all of these little blue things are spores, and those spores get dispersed. Okay, so there's spores now floating around, and those spores eventually reach another somewhere. And if they like that situation, you know, you're a spore, you're like, okay, this has a lot of nutrients. Then they start to form their own mycelia, and then they start to grow there too. So it's like super weird to see a bacteria do this because for the most part, only fungi do this, but there are some bacteria that do it. So yeah, very interesting. Okay, so that's pretty much it for this lecture. Thanks for watching, and I will see you in the next video.